Hello, my name is Kevin Doyle, and I'm doing a PhD in Dr. Campbell Gourlay's lab in the Kent Fungal group. And so I work with a yeast called Saccharomyces cerevisiae, which is better known as baker's yeast or budding yeast. And it's a really powerful tool to study genetics and cell biology with. So my project involves using baker's yeast to try and understand the disease ALS, which is known as motor neuron disease, or amyotrophic lateral sclerosis. So ALS is a very deadly motor neuron disease that involves the progressive death of motor neurons, and it's upon onset, it's really devastatingly quick, and the prognosis is very bad. And so to this day, there's still been no cure, and we still don't really understand the mechanism of how motor neuron disease arises in cells. So a small subset, most of ALS cases are called sporadic cases, so they can happen spontaneously in ways that we don't really understand in a stepwise manner. It could be environmental factors, it could be things that you're exposed to in life. But a small subset of ALS cases are known to be caused by a gene that's passed down in the family. So these are called familial ALS cases, and this is what I'm interested in studying in the lab. So that means that there's a gene that's passed down from the parent, a mutation in a gene that causes ALS. And so the first gene that was discovered to cause ALS in this way is known as SOD1. So it means superoxide dismutase 1, and it stands for, and it encodes an enzyme that basically breaks down oxygen-free radicals, so superoxides, and converts them into hydrogen peroxide and water, which can then be further broken down. So it's basically one of the cell's first lines against antioxidant defense. And it was found that mutations in this gene cause motor neuron disease in people. And so since then, there's been a vast amount of research looking into why mutations in SOD1 cause ALS. But we still don't really understand enough to be able to develop a cure or good treatments for it. And so in the lab, we want to use yeast, which is quite far away from human cells, but a lot of the key biological pathways are conserved between yeast to humans, and a lot of genes like SOD1, for example, are very similar when you compare them in yeast to in humans. So the idea we had was to mutate the yeast SOD1 gene and study the cells. And what we're finding is that when you mutate the SOD1 gene in yeast, the cells become sick, they lose the ability to regulate their metabolism, and the homeostasis in the cell is lost. We think that this centers on the vacuole in the yeast cell. And so the vacuole is basically the storage compartment of the yeast. So it's where it goes to break down old damaged proteins. It takes in toxic metals and compounds and breaks them down. And it basically keeps the cell balanced and ticking along. And what we found in yeast is that when you mutate the SOD1 gene, the vacuole becomes dysfunctional. And so the cell is unable to regulate this homeostasis, which leads to a number of downstream effects like cell death, mitochondria dysregulation, and a number of other factors. And so my PhD is involved around trying to understand further how mutations in SOD1 cause vacuole dysfunction. And what I found is that SOD1 actually interacts with this complex on the vacuole membrane called the VATPase, which is responsible for pumping hydrogen ions into the vacuole and keeping it acidified, which means that it can function properly. And so my PhD is involved investigating this interaction between SOD1 and the VATPase and trying to understand why it is useful for the cell. Yeah, and this is basically what I've been doing for my whole PhD, using the yeast model to try and shed light on new interactions with SOD1 and different proteins in the cell, which could hopefully be helpful to develop treatments and hopefully a cure one day for motor neuron disease. Thank you.